Okay, maybe we can start now. Okay, let's welcome our speaker, Autumn and Manga. They are both research scientists.
I'm sorry, I think I muted myself.
Let me see.
Can you hear me now?
Yes.
Yeah.
Yeah, just very quickly a couple of words about the group, right? It's thanks for inviting us. Very happy to be here to share our research with this privileged group. I think a little bit about our background. We are from Johnson & Johnson Innovative Medicine, right? And the group is imaging and geometric technologies for drug discovery, development, and delivery. So it's sitting under data science and digital health organization. And what we do is to research, develop, and deploy the cutting edge technologies in computer vision, large language modeling, and geometric deep learning to help drive the efficiency and precision and the success across the Geordie pipeline. So I look forward to engaging discussion and set for feedbacks during the presentation. Thank you.
Absolutely. Thank you very much for that information. And with that, we want to welcome everybody to the 7th edition of the Multiomics Reading Group. And with that, we'll be having Arden Moskalev give the presentation, and as well as Mangat Prakash. Please, guys, take it away.
Yeah, hi everyone. I'm Artem. I will talk about our recent iClear paper titled Beyond Sequence, Impact of Geometric Context for RNA Property Prediction. And first, I want to highlight that this is a team effort, and most of the work here was done by our amazing intern, Junji, who really put a lot of effort into making all of the experiments, writing the paper, and then rebuttaling this paper, so it's finally accepted. So kudos to Junji. Yeah, right. So here, what we're going to talk about. First, I will give a brief introduction to RNA modeling, and I will discuss challenges of comprehensive in silico RNA modeling. Then I will give the overview of the data and models that we used in this paper. And then Mangal will talk about experimental results, how we test the models with respect to performance/accuracy, how we test their robustness, and how we test the generalization of our models. Please feel free to interrupt me at any point if you have any questions.
Yeah, actually, could I interrupt already? Just
sure.
to remove that, because you're sharing your screen, there's that little notification bar towards the top center of our screen. So.
Okay. Is it gone?
Yep. Thank you.
Yeah, perfect. Okay. So in this work, we're interested in RNA. And RNA is a large molecule which consists of nucleotides. There are 4 of these nucleotides, and these nucleotides are by themselves are small molecules from 50 to 60 atoms. And RNA, as any molecule, can be represented in multiple ways. So first of all, we can treat RNA as just one-dimensional sequence. That is to say, RNA is just a sequence of nucleotides. But more than that, we can represent RNA with its secondary structure. So RNA has secondary structure. And in that case, RNA can be represented as a planar graph. Also, because it's a molecule, it can be represented just as an atomic system in 3D. And all of these representations are valid for RNA molecules. And RNA, it has very important role in our organisms and in regulating cellular processes. There are multiple types of RNAs and multiple roles that they perform in our bodies. So just to give you a very, very brief overview, so RNA, for example, messenger RNA is important for protein translation. So messenger RNAs are the guys which are used to translate DNA information, genetic information to proteins. Those are called messenger RNAs. And then there are also non-coding RNAs, which either assist certain cellular processes, for example, this housekeeping non-coding RNAs, which assist protein translation, but are not directly translated to proteins, or other non-coding RNAs, they perform some regulatory activities in our bodies. For example, they're switching they can switch on and off certain genes, or they can modify the behavior of protein, and there are really a lot of roles that they play in cellular processes. And actually, the diversity of the roles, of biological roles that RNA plays, it also dictates the diversity of properties of RNA that we're interested in practice. So what do we want to know about RNA? So here are just a few examples of the properties that we're interested in. For example, we're often interested in regions of RNA. For example, in messenger RNA, there can be coding region, there can be signal peptide region, they can be untranslated regions, and even within coding regions, there are multiple subregions. And often it is important for us to know which of the region is where, and sometimes we don't have this information straight away. So we need to predict or to model this information somehow. Then also we can ask about expression prediction. That is to say, it's also about messenger RNAs. Given some amount of messenger RNAs, how much protein is expressed through these RNAs. We're also interested in splicing site prediction. The splicing is a process which modifies RNA of a body, and it can take one form of RNA and spits out another form. So often we want to know how exactly it modifies our RNA. And all of these properties you can also measure in vitro with some type of profiling, which is transcript profiling. But of course, in vitro, that would be expensive, but also it would be slow. Then also other properties, reactivity prediction, stability prediction, or modification site prediction, which are important in vaccine design, or switch modeling and sequence optimization, which are important in manufacturing, actually, when RNA is a part of manufacturing pipeline. And for all of these properties, again, you can use traditional methods like profiling, probing, assays to understand these properties, but it's pretty time consuming and it's very expensive. So ideally, what we want to have a model which takes an RNA and then it predicts one of those properties. And it actually looks like a very simple setup. RNA is an input and we have labeled data. We just train our network to predict this property. But actually, when we started to work on that, we immediately realized that there are so many challenges and so many design choices that you need to make even with a simple problem formulation. And those challenges arise on both input data side, on the output label side, and also, of course, the main question is, given this input and output data challenges, which I will discuss later, how do we select appropriate model for these types of challenges that we expect? So, for example, for the input, usually in academic setting, for example, we have clean benchmarks where we already have the data, and we expect this data to be clean most of the time, and we just use this data to train our model. But in practice, we often need to firstly acquire this data. And then the first question is, how do we get these sequences? RNA, usually we get it from the sequencing platform. And again, sequencing RNAs can be pretty expensive. So the next question is how much of this data we can get. And if we get this data, how noisy this data is, because the sequencing platforms, they are not perfect. They can make sequencing errors. And the sequencing errors, they can have pretty big impact on model performances. Mangal will tell later. Then the next question is, if you remember, I told you that RNA has multiple representation levels. So you can present as 1D, 2D, or 3D. And again, do we have the structure straight away? Like usually in most of the cases, we don't have the structure, but we can predict the structure. And then if this is not a ground truth structure, but if it's predicted structure, is it still helpful? Is it still useful? And how about, for example, if we predict the structure, but we expect some level of noise, how this noise will affect the structure, and will the benefits of having predicted structure still outweigh the drawbacks of noise in this case? And another question could be, when we train the model and we further deploy this model, and we, for example, change the sequencing platform, the noise distribution evolves. So you can say that there is a batch effect now, and how well the model will perform on this batch effect out of distribution, even into account all of the challenges that we described earlier. And the output level, it's also, we face a lot of challenges. One of this challenge is, well, how much labeling can we actually do? And can we afford and can we do complete labeling? Because in many practical problems, we don't have, for example, for nucleotide sequence for RNA, sometimes we want to predict property for each nucleotide. For example, even predict stability, reactivity. We're interested in reactivity for each nucleotide. But in practice, we can't always measure this property for, or let's say we can measure this property, but it's just very expensive, so we don't. So we only have this property for a few nucleotides in the beginning of the sequence. And then the question is, okay, how well the model generalizes on the rest of the sequence when it only sees partial labels. So as you can see, property prediction with RNA in practical conditions, it's not an easy task. And although it looks in practice, although it looks simple, in practice you face a lot of these practical challenges and design choices that you need to deal with. And so when we started to work on RNA property prediction, we also started to look for some studies that maybe provide more comprehensive analysis of how well different machine learning methods perform in the presence of those challenges. And to be honest, we didn't find a lot of guidance and a lot of information in the prior work that addresses challenges. And that's the reason why we decided to do this paper. So to investigate various families of machine learning methods for these practical challenges and to provide some guidance which machine learning methods to use and which machine learning method, what are the pros and cons of different machine learning methods with respect to those challenges. And this list of challenges actually grows almost infinitely, but in this work, we only chose to focus on 5 key challenges that we call tasks in the paper. So there are 5 tasks. So the first one is the impact of 2D and 3D structural information on prediction performance. And that's important to remember that this 2D and 3D information is predicted. It's not ground truth because we don't have ground truth in practice most of the time. So we predict it and it can be inaccurate. Task number 2 is model efficiency with limited training data. You can call it data efficiency. So which of family of the models converge faster in the presence of limited data. Then task number 3 is, let's say we have a lot of data, but now we have only partial labeling. So the first part of the sequence is annotated and the rest of the sequence is unannotated. So again, which model would perform the best in this case? Task number 4 is robustness to sequencing noise. So as I said, the sequencing platforms, they can be inaccurate and they can provide sequencing noise in a primary RNA sequence. But then the question, okay, how does it translate to performance? And how does it translate for the models which work with one-dimensional RNA or with two-dimensional representation, with three-dimensional representation? We also studied this. And task number 5 is generalization out of distribution. Let's say when you train in one conditions and then the deployment scenario, it changes, and then when the model operates, let's say the distribution shift happens, you change the sequencing platform. So the data becomes out of distribution. So which model, again, performs the best in this scenario? So here are the tasks that we use to test our models. Let's now discuss what models are we using. So as I said, RNA, we have 3 types of representation. First of all, RNA is just a sequence of nucleotides, and hence you can use sequence models. So those are usually based on transformers. That's all, not all, but most of these methods are bird-like transformers. You can train it from scratch. if you have labels. So if you have not a lot of labels, you can also use some pre-trained foundational models, for example, RNA-FM or SpliceBird. So these are the models that you use when you work with RNA as one-dimensional sequence. When RNA is two-dimensional structure, it means the RNA is planar graph, and hence we use graph neural networks. This family of models include graph convolutional neural networks, graph attention networks, graph transformers, also the recent models which performed very well on many benchmarks is graph GPS. Those are all spatial graph methods, but also you can do spectral graph methods like ChebNet, for example, to process graph in the spectral domain. And also we added very simple baseline, which we call transformer 1D, 2D, which is just a transformer model. It's a sequence transformer. But for self-attention, we additionally add adjacency metrics to modulate self-attention with the secondary structure. And in this case, it's a method which utilizes secondary structure of RNA. And when you work with 3D, in that case, RNA is a complete atomic system, which can be represented as coordinates of atoms. And hence, you also need to represent the symmetries of the input space that equivariance to translation and rotation. And hence, in this case, you use equivariant geometric neural networks, for example, SNET or EGNN, or use frame averaging or something like DiamNet. So all of these models you can use to process RNA data. And it's very challenging, actually, to compare such a diverse set of models, because each of this model and each of this family of models, they come with vastly different hyperparameters. They also require different training. The training dynamics is different. And it's very hard to compare them like apples to apples. And for that reason, for each of the model on each of the data sets, we fine-tune hyperparameters. And by that, I mean we fine-tune the architectural hyperparameters, so like number of layers, hidden dimension, local neighborhood for 3D methods. And we also fine-tune training hyperparameters, so like weight decay, number of epochs, learning rate. And we do so for each method and for each data set. So that's, now we discussed the tasks and the data, the tasks and the model. And the next question is the data that we use to test these models on those tasks. And the data here is another challenge, because in practice, there are very few data sets that provide RNA property prediction data with annotated structures. So here, actually, we also had to do some work to collect and harmonize the data to make comparison fair. And here, in this paper, we use 4 data sets. These data sets were introduced in prior work, but I think only one of these data sets, only COVID open vaccine data set, it had secondary structures. And the rest of the data sets, they didn't have any annotated structures. So the first thing that we had to do is we had to provide 2D and 3D structures for each of the sequence, each of the data sets. And for 2D structure, we run EternaFold, which is a recent and actually pretty well performing, I would say very well performing method for secondary structure prediction. And then for 3D structure, we use RawFold, which is also very recent RNA folded method. And we run it for every sequence, for every data set. And let me maybe give you the overview of these data sets. So now in each of these data sets, you have RNA as 1D sequence, as 2D planar graph, and as 3D atomic system. These data sets, they cover a wide range of tasks and wide range of properties that you're facing when dealing with RNAs. So for example, this open vaccine data sets, here you predict the degradation profile per nucleotide. So that's for each nucleotide you make a prediction. And you have 4,000 RNAs here, like moderately long RNAs, 100 nucleotides, which roughly equates to 8,000 atoms. For eBonanza, you predict the stability profile You also do it per nucleotide, so for each nucleotide you make a prediction. You have 2,000 sequences, slightly longer, 120 nucleotides, and it's roughly 11,000 atoms. So the next two data sets, they are property prediction data sets for the whole sequence. So you don't make prediction for one nucleotide, but you take the whole RNA and you predict one property for whole RNA. And for TC ribose switches, you predict the switching factor of RNA. And this is a challenging data set because it only has 350 RNAs, which also are small RNAs. And this data set also simulates practical scenario when you don't have a lot of data and you want to do modeling in a very data efficient way. And another data set is fungal, where you predict the protein expression for messenger RNA. It has 7,000 RNAs, up to 3,000 nucleotides, so it's also very long sequences. And when you predict 3D structures of these fungal RNAs, it actually takes a lot of time and it roughly contributes to 200,000 of atoms. Actually, running this 2D and 3D structure prediction models with this fungal dataset was a challenge by itself. And so that's one of contribution of the paper, that we provide these 4 curated datasets annotated with 2D and 3D structures. And not only we provide this dataset, but we also benchmark a lot of methods on these datasets. Now I think I will hand it over to Mengel.
Thanks, Artem. Can you still hear me well?
Yes.
Okay. Yeah. Next slide, please. So as Artem described, we start by asking the question if adding the 2D and 3D structure improves performance essentially over sequence-based models. So as was previously mentioned, that these structures are not ground truth structures, but they come from structure prediction tools. So the question is essentially that then, which means that they do have some errors as well, because the structure prediction tools for RNA are not as reliable as those for other modalities like proteins, etc. So the question is, even using these structures, which are coming from structure prediction tools, can we gain some information which is useful enough for downstream task performance? And this is the first task that we look into. So here we compare 2D models, which were described earlier, with 1D model first. And we already see that the 2D models, and we show here the RMSE, the lower the better. So what we see that even adding the 2D information essentially helps improve the performance over the sequence-based models, even compared to some foundation models like RNA-FM and SpliceBird. And we also find that the spectral graph neural network, which is the SHEBNet, outperforms the spatial GNNs. And this is likely because it can capture global graph features right from the first layer, enabling a global receptive field, whereas spatial GNNs aggregate node features layer by layer, essentially, and they mostly focus on local neighborhood, essentially. This is also resonates with what we know about RNA, because RNA is a large molecule, and essentially different parts of this large molecule may interact with each other to influence the property downstream, so to say. So if a model can reason over longer context, it's usually better. And so yeah, it's along somewhat expected lines that we see that SHEBNet seems to perform better. But in general, the 2D models seem to outperform the 1D models. Next slide, please. Yeah, and then if you look at 3D models, though, which reason at all atom level, although they have all the information that is in the 2D representation and the 1D representation, we find that the 2D models do not perform that well. They're actually even worse than the 1D models. And there are 2 hypotheses to it. Next slide, please, Artem. So one is basically that the 3D models by default have limited receptive field. And so they're not able to capture all the relevant information that is needed for the downstream property prediction tasks. And the second hypothesis is the 3D predicted structures themselves, since we are using the 3D structure prediction tools to get the structures, they're not so accurate to begin with. Hence, essentially modeling them directly does not seem to bring much benefit. So let's now also look into these hypotheses one by one after the other. But also another interesting thing to note is that for larger data set like fungal, we see that the 3D model operating on all atom resolution simply runs out of memory. So that's also another limitation of the 3D models. Next slide, please. So the thing is essentially, we know essentially that we can naively also increase the receptive field of these 3D models, so to say. And if we add more layers, essentially, then we can reason over, then we can have a larger receptive field. But the problem, as we already saw in the last slide, even for larger data sets, is already running out of memory, so to say. So increasing the receptive field naively, essentially by increasing the local neighborhood, does not scale well on larger data sets, and especially relevant for large molecules data set like RNAs. So we propose a simple approach for efficiently scaling the 3D models while maintaining all atom resolution. So what we do is we start with all atom graphs, and the initial few layers of our model, essentially, they operate on the all atom graph. Then, because we know essentially that a nucleotide is composed of some number of atoms, essentially. So what we do is we do a pooling of the features of the nodes corresponding to the nucleotide after some layers, essentially. So after some layers, our model is operating on the nucleotide pooled graph. So the benefit of this is that this reduces the graph manifold, and it makes it easy to scale, but also maintains at the same time all atom resolution in the initial few layers. So with this combination of all atom graph as well as nucleotide layers, essentially, we are able to process larger graphs and have an effective larger receptive field. So the question is, does it improve performance anyhow? So next slide, please. So here in the very last category, we see that these are 3D models with nucleotide pooling. And we do see that there's remarkable improvement over the 3D models which were operating on all atom without any pooling. So this showcases that essentially our hypothesis of 3D models having limited receptive field was correct, essentially, and increasing the receptive field seems to benefit. But still, we see for that the performance of the 3D models still lags behind. It improves upon the 1D models already now, but it still lags behind the 2D models. And this relates to a second hypothesis, that this is likely because the noise, there's still a lot of noise in the 3D structure prediction tools, essentially, and hence the 3D structures that these models are operating on are not so accurate. So for example, if we compare for a single sequence, the structures predicted by these two, by a set of two, by a pair of two structure prediction tools, qualitatively, we can see there's a lot of difference between the different parts of the structure predicted by the different tools. And also, I mean, to put it in numbers, essentially, the difference in RMSDs between any of these pairs was ranging from 14 to 40 RMSD, essentially, which is a lot. So, which also goes on to show that the 3D structures from these structure prediction tools are too uncertain, too noisy, which of course also makes sense why 3D models essentially operating with these structures do not work as well as 2D models. Yeah. Next slide, please, Artem. So in the next setting, we investigated the task 2 and 3 that Artem introduced earlier. Well, the first setting is the limited data scenario, where for each RNA sequence, we do have some property labels available. But there's lots of data where we do not have, such as the ones shown on the right, where we just have the sequences and their corresponding predicted 2D and 3D structures, but we don't have their property labels available. Next slide, please, Artem. Yeah, and then the other scenario is a partial label scenario, where, as described earlier, in many real-world settings, essentially, we do not have, if it corresponds to the nucleotide level labels, we might not have essentially full labels available for all parts of the sequence. So we might have labels only measured for the initial, let's say 20-30%, and we might not have the full labels corresponding to the other parts of the sequence. Yeah, next slide, please, Artem. So yeah, these basically show real-world settings, in the first setting where we have scarce RNA datasets with labels, because it's costly to acquire, which means the models will have to learn effectively to learn from small datasets. And in the second setting, we have partial or incomplete labels due to the high annotation costs, so to say, or experimental costs, which also necessitates that the models learn to generalize from partial labels for the regions where we do not have the labels available. Yeah, and put in machine learning terms, essentially, I mean, this is the scenario that we have on the left side, the limited data scenario, where we have in the training set different fraction of the training data for which we have the labels available. And on the right, we have the partial label scenario for which each of the sequence essentially in the training set we only use the labels for a part of the sequence. Yeah. So let's see how well do the models work in these two settings. Next slide, please. Yeah. So this is just a very high-level comparison of all these models that we discussed earlier. On the x-axis, we see the fraction of training data or the fraction of label data that has been used for training. And on the y-axis, we see the root mean squared error. The lower, the better. And in green, we see the ShebNet again. The 2D spectral method seems to outperform all the methods for limited data regime or for partial label regime. And we see that the transformer models essentially, which is shown in the pink, seems to be very data hungry, which is not surprising. And it's similar things have been reported in other fields as well. And they need 2 to 5 times more data to match the performance of even the least performant 2D models. Interestingly, here as well, we see in low data regimes, essentially 3D models seem to outperform 1D models, despite the structural noise that is coming from the structure prediction tools. And additionally, we see that even the simple extension of the transformer 1D, as Artem described, transformer 1D 2D, which takes into account some topological information, seems to perform much better than the transformer 1D model, which operates on sequence alone. I think there's a question from Cesar.
I think that is a very, very interesting result, right? Despite having that structural noise, would you say this is coming from some sort of implicit codon bias that is still included in the 3D structure, but perhaps doesn't carry over as reliably when you have only sequence or something similar? What is the biology here, in other words?
Yeah, could you repeat, Cesar? I think I missed the last part of your question. You mentioned that could it be because of the intrinsic codon bias, and then you said something which I missed. Sorry, my internet is somewhat poor. Could you repeat, please?
That's right. Yeah, that's basically the gist of the question. Despite having structural noise in the 3D model,
yeah.
What do you think is the biology between these? Like, is it just a codon bias that's being supplanted in both cases? Like the 1D models have sequence, right? 3D models as well. But do you think it's just like implicitly in the 3D structures or not? What is the actual biology? Why are we getting an outperforming model like considerably?
Okay. Yeah. So potentially, feel free to pitch in, Artem, essentially. But let me take a stab at this. I feel essentially 3D structures inherently have a lot more information, right? I mean, than just the sequence alone. And depending on what level of noise we have, essentially, as we'll see also later, what level of noise is good enough essentially to still eke out any useful information from the models. I think that's part of the reason. I'm not entirely sure if it's anyhow related to the codon bias or not. And it's an interesting hypothesis, but I cannot say for sure if that's the reason. It's just that the 3D structures, even though they're noisy, they're probably not that noisy that it just destroys all possible information that is in there. And just because they have a lot more information than just a 1D sequence alone, I believe that's why it works. And later in the later slides, we will also discuss essentially the impact of noise. So how much noise can these models tolerate in the first place, essentially, and what? And that might help partially answer your question as well.
I would also add that these RNA folding methods that predict 3D structures are much less advanced at the current stage than methods for protein folding. So it's just maybe we're also lacking very strong methods for 3D structure prediction for RNAs. And it's also partially because the data availability for RNAs, I think it's more challenging than for proteins.
I like those answers. Thank you very much.
Thanks. Yeah, and hopefully in the next, when we look at the next settings, essentially this will become a bit more clearer essentially on in which setting should we under what kind of noise should we expect the 2D models to work better or worse. Yeah. So as we were just talking about, so there's not just one source of noise. So the one source of noise is coming from the 2D and 3D structure prediction modules themselves. But of course, sequencing errors are also common. And there are different kinds of sequencing errors: nucleotide insertion, deletion, or mutation. So here, for this purpose of this study, we only considered mutation essentially, which means a nucleotide, for example, C in this original figure, the second one has been, the third one has been mutated to G. And this depends on essentially the sequencing platform used and the quality of sequencing that has been done. So what we did was essentially we looked at the sequencing error rates from different sequencers, as has been reported in previous literature. And then we tried to replicate these sequencing errors to mimic the real-world sequencing platform failure cases. Yeah, next slide, please. And then we asked the following two questions, or the tasks. So how robust are these models to the sequencing noise? For example, if we have in the training data, which has been coming from one sequencer which is noisy, and if we test on the similar kind of on the data acquired from the same sequencing platform, which has almost the same rate of or probability of sequencing errors, essentially, how well do these models perform? So notably, essentially, to the labels, we do not introduce any noise, just because the labels are being measured separately. So once we have the RNA in vitro, we can measure always the labels or the properties separately, independent of the sequencing platform. So there's no noise introduced to the labels, only the sequence. There's errors in the sequencing data, and this aims to simulate the case essentially, that the models have to be resilient in real-world applications to ensure the reliability. And the second scenario is the generalization to outer distribution noise, where we have the training data, which is let's say acquired from a high quality sequencing platform. And now we have a model which has been trained on this data and it has been deployed in production. But of course, imagine that in the future, we have data coming from another sequencing platform which might have errors, or essentially we have some data from the past, essentially, which we acquired from another sequencing platform, and we want to run it through this deployed model. So how well do our models generalize to unseen conditions during deployment is what the second task is trying to simulate. So next slide, please. So here on the left, essentially, we see the results for the task 4, which is the robustness to noise. And on the right, we see the generalization setting to OOD data. And again, on the y-axis, we see the RMSE, lower the better. And on the x-axis, we see the noise ratio. So how much sequencing error has been introduced on the sequencing side. And of course, this sequencing error, of course, accumulates through the 2D and 3D structure prediction tools as well. So not surprisingly, we see that the transformer 1D models, which are only reasoning on the sequence level, perform the best in this scenario, whereas the 2D and 3D models perform the worst actually in this scenario. And partly to answer the earlier question, essentially, if we increase more and more noise, then of course we can imagine that the structure prediction, that the errors from the structure prediction tool gets compounded by the sequencing noise as well. And hence, at some point of time, and essentially we end up having very little information in the 2D and 3D structures, which is informative of the downstream property prediction task, essentially. So this is not a very surprising result in that sense, because we see that 1D models are more robust under sequencing noise, because they only have to deal with the sequencing errors, whereas the 2D and 3D models have to deal with not just the sequencing errors, but the effect of the impact that this sequencing error has on the predicted structures downstream. So to illustrate a bit further, could we go to the next slide, please, Artem? Yeah. So just to showcase essentially what we mean. So on the first column, if you see, there's no sequencing error. So this is how the 1D sequence, 2D structure, and the 3D structure look like. If we mutate the sequence with each nucleotide having a probability of mutation of 0.1, essentially, we see only with this amount of sequencing noise, the secondary structure and the 3D structure change dramatically. And by the time we get to about 0.3 probability of mutation, so to say, you already see that the 3D structure does not even resemble anything to what it was in the beginning, so to say. So also going back to the last question, this is what I was meaning essentially. So at one point in time, if we have more and more noise, essentially, then there's not much information in the 3D structures for the models to do anything meaningful out of it, essentially. And yeah, so next slide, please. So basically, in summary, what we just talked about, that RNA can exist as 1D sequence, 2D structure, and 3D structure. And most of the studies so far have looked at just using 1D sequences to model RNAs, and there's lots of models for that, including foundation models. But 2D structures and 3D structures have hardly been explored for property prediction tasks. And this is partially because there's no high quality structural data available for RNAs, unlike for proteins, with annotated property labels. So here we tried to ask the question, if we use 2D and 3D structure prediction tools, which we know are not as reliable as those in other domains like proteins, can we still do something better by modeling just the 2D and 3D structures than just by modeling 1D sequence alone? And then we looked at different kinds of models, the 1D models, 2D models, and 3D models, and provide general guidelines on essentially what to expect in which scenarios from the partial data or limited data settings, and also in robustness and to sequencing noise and generalization to outer distribution noise during deployment scenarios, essentially. And we saw that adding the structural information seems to help, and especially when the data is limited or the labels are limited. But the 1D models are more robust to sequencing noise, which the 3D models, while the 3D models are the least robust to the sequencing noise. So yeah, this is our first investigation, essentially, along these lines to understand what can be done using looking at all the three different representations of the RNA. And I'm sure that, yeah, this is only the first step, essentially, and going forward, researchers essentially will probably pay much more attention to this kind of to this domain. And then, yeah, I'm keen to see what comes next. Yeah.
Yeah, that's also our last slide. Thanks, Mango.
Thanks, Artem. Thanks, everyone.
That was a great presentation.
Thanks.
So now we're gonna take I guess we saw little like hand claps and confetti on the screen, which is a way to thank your speakers. So with that, we open up our Q&A session. If anybody has a question, please write it in the chat or feel free to open up the mic.
So maybe I can start with one.
Absolutely.
Yeah. One thing I wonder, when you do these prediction from 1D to 2D and then to 3D, you feed into your neural network only the 3D structure or only the 2D or only the 1D, right?
Yes.
Yeah, and then for 2D structure or for 3D structure, for example, we have like node features, and we have topology, and node features are in 2D case, that's nucleotide identity. In 3D case, that's atom identity, or after nucleotide pooling, it's also nucleotide identity, plus the connectivity that you have from the structure.
Yeah, have you considered to consider that as a multimodal input? Because they are just projection of one another.
You mean using them all together? Yeah, we have considered that. Yeah, Mango, do you want to share some words on this?
Yeah, so we looked into it essentially, and then we tried to combine all these three representations together, And we know already that seems to work better than just looking at any of these alone. But that work is still not out. So we know that essentially that the natural extension would be to consider them all together. And the initial suggestions are, and the initial results seem to suggest that essentially putting them all together seems to do better in all the settings that we discussed. And then, of course, the right way to combine is up for research as well, right? And hopefully soon, we'll be able to share a little bit more about it.
Okay, thanks.
So that makes me immediately think, you probably can figure out a way to quantify like how the noise is doing in your data, right? So in that multimodal objective sort of, maybe there can be a way to use, if you're very confident about your 1D data, you could use that to somehow refine your 2D data, if you have like some sort of commonalities between these, so on and so forth, and sort of anchor the data where you had the most confidence that was less noisy, let's say. But
that's a good point. Indeed, we have this knowledge prior to deploying of a model. I think that's a good suggestion. Yeah, we can weight the modalities according to expected noise level. I think not in all scenarios we know beforehand what is the level of the noise in sequencing, although in many scenarios we do that. In this case, I think also if we just have the data set, the model can learn it from the data if it's consistent, to kind of rely on one modality more than on other modalities.
Yeah, of course. And having that on the forefront of like the experimentation pipeline, I don't think most are aware about noise being a considerable thing as far as models. But yeah, wonderful. Dylan?
Yeah, so you had a few slides showing how sensitive, especially the 2D and 3D predictions were based on simple like nucleotide errors, for instance, base errors. First of all, I don't know how, like, at what point is that acknowledged in the field that these prediction models are highly sensitive to these kind of sources of noise? So it seems like there's like a robustness issue, maybe. I want to echo like the previous question about like if you can kind of leverage the more robust properties of the 1D models to then inform the 2D and 3D models, right? So the 1D models, you're more robust, right? So is there a way to use those as a way almost to denoise or robustify the input signals that are then used for the 2D and 3D models?
Totally. I think what you can do is you can use this 1D models to initially extract the nucleotide features and then use these nucleotide features inside of like 2D topology. You can do something like that. But then also, noise is a very important aspect, but it's not the only one aspect. So one of the things about 1D models, indeed they're the most robust to noise, but they also require the most data to converge. So we've seen that usually the amount of data for a simple sequence transformer to converge to the level of ChapNet, you need 5 times more data. You can still converge to the same quality, but with the 5 times more data. And that's in practice, we don't usually have that much data to afford ourselves that type of training dynamics. And this is why we also want to include as much prior knowledge, including secondary structure, even if it's noisy a little bit, to facilitate this convergence. But yeah, I think that's a very potentially fruitful line of work of how to properly combine those modalities. And yeah, again, I agree with Manuel. There are a lot of ways how to do that. I think, yeah, if it answers your questions.
Yep. Okay. But there is a data issue for the convergence issue rather for the 1D models.
For 1D models, yes. If you don't have topological structure or 3D structure, you need more data to converge.
Okay. All right. Thank you.
If we still have time. So you in the graph or sensitivity in the 1D data, you considered error rate of 10% and 5%. These were the first point.
Yeah.
where you look at the. I'm wondering why you decided to use such high error rate when sequencer nowadays do 0.1% error rate. This is kind of standard. And if you consider that you would be resequencing multiple times the same RNA, the standard is to go for 30x in measuring the mRNA. So you have close to a null chance of seeing a sequencing error there.
Yes. I'm not an expert on sequencing platforms, but from what I understand, it heavily depends on the sequencing platform. So you probably refer to Illumina or Illumina sequence platforms, which are can be pretty reliable. But if you use, again, I'm not expert, so I don't want to say something stupid, but I think Oxford sequencing platforms, they're much more prone to noise, and the numbers that we put on the paper, something that we found in existing literature that's saying that these sequencing platforms, especially the Oxford sequencing platform, they can go up to 30% noise. But I agree with you, if you have very, very good sequencing platform, which is only 1% of the noise, then yeah, there is not so much problem with noise in this case.
Yeah. Just as a comment for the field, is the fact that very often when we do single molecule sequencing, as with Oxford nanopore sequencing, we do on top of that an Illumina sequencing, just to solve the problem of error sequencing, and it's not that costly.
I see. Yeah. Well, that's a useful feedback, essentially. Yeah.
Can I go for another one?
Oh yeah, like we started
Okay.
like at 11:05. We can go for 5.
Okay, cool. When you consider the 2D structure, do you consider, so 2D structure is a weird thing in RNA because it was defined algorithmically in the 80s. But the fact is that secondary structure often contains pseudonodes, which is non-hierarchical base pairs. Does the method that you use for obtaining the 2D structure consider the possibility of pseudonodes?
I think it does. So I don't remember exactly, but I do remember. So what we did was, for the 2D structure prediction, the reason we took the prediction tool that we utilized was because it was reported in a recent Nature Methods paper from Stanford, the Open Vaccine Eterna Fold Challenge. And I think the method that they used essentially was considering the pseudonodes.
Okay, nice.
But I might have to double check.
Yeah, well, I'll double check. And the other aspect of secondary structure that is not considered very often is the possibility to have non-conventional base pairing, which happen all the time in 3D structure of RNA. Like you've got the GU, which is sometimes taken into account, but after that you have GA that is also quite frequent, and quite a few others.
Right, so all still possible with that. So in the interest of time, I think that finalizes the session for today. Thank you very much to our speakers for coming again, and this finalizes the recording. Thank you very much.